So one of the speakers in today's session is not shown up, Dr. Patrick Liu, so unfortunately we won't hear from him. But I welcome the next speaker, Dr. Lloyd Johnston of Selecta Biosciences. He's going to talk to us about something different, using vaccine particles instead of inducing immune response. I think what he's going to talk about is immune tolerance. So, thank you.
So I think I'm waiting to bounce across the screen from Monty Python now for something completely different. Nothing related to what has been going on this morning, though it's been very interesting. I'm going to talk about, as Neil said, antigen-specific immune tolerance, and the key here being antigen-specific. So not global immune suppression, but antigen-specific immune tolerance. To tell you first a little bit about Selecta. So we make synthetic nanoparticles. We call them synthetic vaccine particles. And we can do a lot of different things with these particles, doing a lot of stimulatory vaccines, but recently we've moved more into doing tolerogenic work as well. And one of the keys with our technology is that inside the nanoparticle, we always encapsulate some sort of immunomodulator. So it's a tolerogenic agent, which I'll talk about today, or when we're doing stimulatory vaccines, we put in an adjuvant like a TLR agonist. It's not just a nanoparticle, but it's a nanostructured particle, so we use a self-assembly technology where we can put certain things on the surface, certain things on the inside. And on the surface here, if I'm trying to do a stimulatory vaccine, I put a B-cell antigen to then generate an immune response to that. And we've done it with small molecules. Omid, one of our founders, talked about our nicotine vaccine in yesterday's presentation, sugars, small molecules, proteins, etc. But what I'll talk about today is, without putting anything on the surface, we'll put inside the particles things to activate the T cell, so either through the MHC class 1 or the MHC class 2 pathway. And we can do this with peptides, just using epitopes, or with using full proteins to engage in that pathway. And the other key is the particle itself. So the particle itself, and as many people who work with nanoparticles, we use polylactical glycolide acid, the PLGA. It's been in people for over 30 years. For those not familiar, if you had resorbable sutures that just the doctor puts in and they disappear over time, it's the same material. In the presence of water, it breaks down the natural metabolites, lactic acid and glycolic acid. It's a very flexible system. If you, in a very simplistic way, think about your lactic and your glycolic acids as, different colored Lego blocks, you can put a lot of blocks, a few blocks, lots of reds, a few blues, and by changing that, the molecular weight and the ratio of the lactide to glycolide, you can get very, very different properties, which can affect things like the size of the particle, the surface charge, and very important that we've found is the release rates of the immunomodulator. So we can make plenty of particles with the same components that work extremely well and a whole bunch that don't work well. Although they have the same components in them, they might have different polymers, been made in a different way. And as I think many of you know here, why do we use nanoparticles? So what we're actually trying to do is just work with nature. So what you hear a lot about is trying to be stealth particles and get them to the site of infection or or We don't. We're not trying to take our particles and get them to the site of infection. Really what we use the particles to do is encode a message or give a blueprint to the immune system to give a response that I would like it to have, either a stimulatory response if I'm trying to raise an immune response, to either protect you with antibodies, or create cytolytic T cells to go kill cancer cells or chronic infection, or in what we'll talk about today, to give in a specific signal to tell the body to be tolerogenic to a specific antigen. So we have a number of significant advantages here. They have very high efficacy because the particles themselves, your immune system's evolved over millions of years to protect you from pathogens, which are by and large nanoparticles. So as you know, viruses are nanoparticles. Flu virus is about 80 nanometers in size. They go down to maybe 30 nanometers with hepatitis B, and they go up to a few hundred nanometers. And bacteria go from a few hundred nanometers up to a micron or two. So we're actually trying to work with the immune system by giving it something that it really wants to recognize. So when we design our particles, we inject them, we're trying to get maximal immune recognition from them. We can do this sub-Q, and in that case they flow to the draining lymph node, or if we give them IV, and with pretty much every nanoparticle, the majority of the particles, and you give them IV, they go to the spleen, they go to the liver, and those are also very good places to have an immune response. So we're actually working with your body to try and give it that blueprint to make what we want. There's a lot of enhanced safety profile here, and particularly on the tolerogenic, on the stimulatory side. So often you give adjuvants, you give them free, they go everywhere. and you get all sorts of off-target effects. We give very small amounts of the adjuvants because they're being very targeted. They go just to the immune cells that we want, the antigen-presenting cells. So we can give maybe 1/10 the dose of an adjuvant and get 10-fold higher increase compared to a free adjuvant. We're not quite at the point of small molecule design with, turning a structure-activity relationships, but these, once you have the building blocks, these are actually quite easy to make. So if you know what you're doing. So a research associate will make 12 of these in a day. So we can do hundreds of formulations, and we then characterize those formulations analytically, and then things that are differentiated we put into in vivo models. So we actually, whether I'm trying to do a stimulatory vaccine or trying to do a tolerogenic vaccine, we have a pretty good idea because we can slightly vary the components. So have a little bit, we can change the density on the surface. We can change by very precise amounts the amount of immunomodulator that's inside, and we can actually sort of know the boundaries where we want to be in when we take a new project on. And they're very relatively simple to produce. So prior to coming, I was actually the first employee of Selecta. I was at Alkermes, whereas VP of Manufacturing, VP of Operations. We had very large plants that made emulsion-based polymers. They're microparticles, but we use all these basically standard unit operations. And what's interesting for me, I've built and run very large plants, 100,000, 300,000 square feet, $50, $100 million. It's actually rather simple to make. With vaccines, you only give it a few times, right? So when you're thinking about scaling up things, if you have to take a drug every day, you have to make a lot of it. But a vaccine you take 2 or 3 times, and in the way we're doing the tolerance, it's like, in a sense, a negative vaccine. We give it to you a few times, and it teaches your body what to do. So this is our phase 1 scale. We completed a phase 1 trial in our nicotine vaccine. And so it's very small. That's about 10 grams. And you typically give small amounts. So you might give a milligram. So that may be close to 10,000 doses. That fits on a benchtop. So very different for me where I've had to build large plants before. But it's very scalable. It's much less expensive than having to build fermentation and other things if you're doing protein-based nanoparticles or just doing proteins for vaccines. So we work in 3 major areas, and what I'll talk about today is our antigen-specific tolerance. And these are first-in-class therapies for us, and one of our current focuses is actually the elimination of anti-drug antibodies. But when we talk about antigen-specific tolerance, what we're trying to do is teach the body to stop having an immune reaction we don't want. So it could be an autoimmune disease, it could be an allergy, which is just an unwanted immune response. But we're focusing on now, and I'll get into why that is, what are called anti-drug antibodies. As we know, biologics are one of the fastest growing classes of drugs, but these are proteins. And a number of people actually, when you get injected them, just like you get a vaccine, which is a protein, they make an immune response to it. So the next time they get the drug, it doesn't work because the antibodies have cleared it. And this happens in a lot of drugs, and very highly, in high frequency, it happens in enzyme replacement therapies because these patients or these babies are deficient in that enzyme. So when I give it the enzyme which it needs to live, it sees it as completely foreign, so it does what your body should do. It makes an immune response to it. So the next time I give it, it just gets cleared because you have a lot of circulating antibodies to that biologic. So what we're trying to do is teach the body not to do that without doing a global immune suppression. And we'll talk about that a little bit more today. We also can do cytolytic T cells, and we have some very interesting data by changing the composition here. Often with cytolytic T cells, what happens is that you can give a vaccine, and DNA vaccines and others, and you get this expansion of T cells over about 7 days and get a rapid decline. And we can do that with our particles. We can get rapid expansion over 7 days and decline. By changing the formulation, we've actually been able to get a rapid expansion and keep it up for over 30 days. And that is also translated in some of the oncology models to better efficacy. In antibodies here, we focus on actually a little bit more in the public health area with grants. As OMID, we have a grant with the Gates Foundation on malaria, and we recently received an $8 million grant from NIDA to continue our work on our nicotine vaccine. So this is our general pipeline. In the blue is our tolerogenic programs. And just last week, we announced that we've licensed a pegsiticase, a uricase for refractory gout. If you have refractory gout, you have high levels of uric acid, and none of the other drugs work. It's an orphan disease. It's a small population, 50 to 100,000 patients of gout, but they basically have these high levels of uric acid, which I, being a chemical engineer, not a biologist, but what I found fascinating is all mammals, with the exceptions of humans and greater apes, naturally have uricase. And uricase is an enzyme that's very effective in chewing up uric acid. So there is a product that's on the market, which is uricase, but over 90% of the people get high levels of anti-drug antibodies to it. So it doesn't do very well because it just doesn't work. It works the first time, and then as they get the next one, it's not doing anything because it's being cleared. They also have high levels of anaphylactic reactions. So we've licensed in that biologic, and we'll combine it with our synthetic vaccine particle to make a non-immunogenic uricase for refractory gout. We also, when I'll show data, well, I won't show any data on this program, but I will show data on our hemophilia program. And if you're a hemophiliac, you don't have factor VIII, or if you're hemophilia A. So when I give it to you, which you need to clot, about 30% of the people make antibodies to it. They're called inhibitor patients. And then the next time you get the factor VIII, it doesn't work. So what we do is we give a few injections at the start of the therapy, which basically teaches your body to be tolerant to the factor VIII. We have a large collaboration with Sanofi on food allergies, and we work on an autoimmune disease with type 1 diabetes. On the stimulatory side, we look at some chronic infections, our smoking cessation, our malaria program. So let's talk about anti-drug antibodies. So the impact of anti-drug antibodies is very big. It neutralizes, so you have no efficacy, so the next time you get it, it doesn't work. You can get alteration of the PK, so it might not neutralize it, but it changes what the PK curve looks like, which actually can change the efficacy and make it not work. And there's a number of hypersensitive allergic reactions, including anaphylaxis, that you can get to these biologics. And these are all well documented in the literature. So there's a large opportunity for tolerogenic therapies with current drugs or drugs on the market. So this enables the treatment of patients that have had immunogenic responses, so you can bring them back in. You can look for what happens. I'll show an example. It's not a drug that we are pursuing. It's more of a proof of concept. But if you look at the anti-TNF drugs like Humira, it was demonstrated in JAM about 30% of people have PK altering, basically loss of efficacy, 30% of the patients because of antibodies. But in that, what happens is if you're giving Humira, it stops working, you take Enbrel. If that stops working, you take Remicade. So you get that to basically a switching that goes on. But you can use this potentially to make a non-immunogenic one and look at life cycle management. There was a talk yesterday on all sorts of a whole session on new biologics. And if you look at these novel drug developments, you start out with some antibodies like Remicade's a mouse chimeric, and you get all the way down to Humira, which is fully humanized. It still has 30% people with anti-drug antibodies. Now we're talking about bispecific antibodies, antibody-drug conjugates. We're getting more and more away from self. So it's likely that these are going to be more and more immunogenic. So for these therapies to be effective, you're going to have to teach the body to be immune tolerant to them, because otherwise they may work on the first dose, but on the second dose you'll have an immune response to it. So how does this work? This is where our synthetic vaccine particle has an immunomodulator in it and an antigen. It goes into a lymph node. And as one of our founders, Uli, says, the dendritic cells are the school teachers of the immune system. And so when it's born, when it's a new and naive dendritic cell, it has a career decision to make. It either becomes a stimulatory dendritic cell and gives you a stimulatory response, or it becomes a tolerogenic. You're mainly biased to this pathway because you're tolerant to yourself, so you have lots of proteins in yourself. But what we do is we put in the immunomodulator. It's picked up by the dendritic cells very readily. And so now that in the context of this antigen, it has a tolerogenic signal that's inside the particle, which then takes a naive T cell and makes it become a T regulatory cell. We've also actually found it deletes effector T cells as well. So we basically bias the immune system for this antigen in an antigen-specific way to make antigen-specific T reg cells. So this is an example just using some model antigens with the antigen specificity. So this is a particle with OVA inside it plus the immunomodulator. We give that on day minus 14. Then on day 0, 14, and 28, we give the mouse an injection of both OVA and KLH, both immunogens. And what you can see with no immunization, these are the log scale titers. This is for OVA, this is for KLH, OVA in black and KLH in red. So if we just don't do any treatment here, you can see what the normal response is. If we just put the immunomodulator in here, but without the antigen, so there's no antigen-specific context, the mice are normal. But if you see the ones that got the OVA plus immunomodulator, their OVA titer is essentially 0, but their KLH is normal. So we've not immunosuppressed the animal. It can have normal immune responses, but we've educated the immune system to say OVA is not an immunogen you need to be worried about. So be tolerant to that. And the encapsulation of the immunomodulator is key. So the immunomodulator we use, in this case it's an approved drug. It's used as immunosuppressive therapy given chronically. But if you don't give it chronically, it actually acts as a stimulant. So we're giving this just the once a week instead of giving it daily. So if we give a nanoparticle with OVA inside, these are looking at antigen-specific T cells, so stimulatory cells. With the OVA plus immunomodulator in the particle, we reduce those. If you give the free immunomodulator, remember it's immunosuppressant given daily, but if you give it weekly, kind of like a vaccine, it acts as a stimulant. It actually increases the number. It actually makes things worse. And then here, these are FOXP3 markers, which are FOXP3 are a marker for T regulatory cells. So here, you've got the nanoparticle with just OVA. Here, if we give the OVA, the SVP with the OVA and the immunomodulator inside, we get an increase in regulatory cells. And the free immunomodulator, again, immunosuppressant, but because you give it on a weekly basis, it's now actually reduces the number of Tregs. So you can see here with the this is looking at just the antibodies with the SVP and then the modulator and the particle. So the adverse effects of the key adverse effects of ADAs are neutralizing, alteration of PK, PD, and hypersensitivity. So I'm going to demonstrate that with a couple of ones. First of all, with just KLH. Again, so here we're giving the SVP treatment IV with KLH IV, and we give it 1, 2, 3, 4, 5 times, and then we can just give KLH another 4 times. The KLH IG antibody levels are here. So when you give no treatment, you get high antibody levels in black, and this is an anaphylaxis score here. So up to on this scale, I think 5 is dead. Here, there's severe anaphylaxis and moderate anaphylaxis. So they're getting anaphylaxis as well. Whereas with the SVP treated group, 1, they don't make any antibodies, and 2, they have no anaphylaxis. So we can treat that particular issue of the ADAs. So I'm going to show 2 proof of concept studies here. And last time I gave this talk, I got a lot of questions of why would you want to do this for Humira. It's not a product that we're pursuing. It's more of a proof of concept. Then I'll show you some of our factor VIII data. So these are transgenic mice that express human TNF-alpha. So they develop arthritis around week 5. So at week 5, as they're starting to develop arthritis, what we do is we treat them with the SVP and with Humira. We do that for, I think it's 7 weeks. And then we stop giving the SVP treatment, and we just give Humira out through 20 weeks. This is the week 17 data. And so what you see here is the arthritis score. And so and the maximum score on this scale is 16. So here, you have these, the PBS mice have bad arthritis. They're being treated with Humira, very good drug, but it doesn't work that well. It is statistically significant, but they still have pretty significant arthritis. But if I give them Humira plus the SVP, they're essentially normal. And what's going on here? Well, what happens is if you just give Humira, because particularly in mice it's even more immunogenic because it's a humanized antibody, you have very high levels of antibody. But here, with the SVP, with the exception of this one mouse, which is still 2 orders of magnitude lower, we basically have no antibodies. So we've prevented the body from making antibodies. And this is all the way out here at week 17, so they haven't seen the SVP in over whatever that is, 6, 7 weeks. And what's happening is we're really normalizing the PK. So this, again, we're giving SVP. This is the Humira is given sub-Q. This is all sub-Q data. And if you give the first dose of Humira, you get this level in the blood, circulating blood of Humira. After the 6th dose of just Humira without SVP, you basically have no Humira in the blood. Why? You have high levels of antibodies, and it's clearing the drug. But with SVP, on the 6th dose, it looks just like the first dose because you haven't made any antibodies to it, as you see here. And there's also local injection site reactions. These are sub-Q. It's in the paw, so you can look at paw swelling. So with no treatment, you get significant paw swelling, and with the SVP, you get very little paw swelling. That's really just from the saline injection, or the volume of the injection that's going into the foot pad. And you can see this in, I'm not sure how well that shows up, but this is week 20 data. So this is a histopathology data. So this is a X-ray. So if you only give Humira for 20 weeks, you get that level of arthritis in the mouse. And the reason is it has antibodies, so those subsequent injections after the 2nd or 3rd injection are just not working. Whereas here, you have in the, it's not working. On the Humira SVP group, you have basically and essentially a normal-looking joint. And you can see that here looking at both inflammation, pannus, cartilage, bone resorption. Humira is essentially normal. I mean, Humira plus SVP is normal, whereas you have doesn't really look any different between no treatment and giving Humira for 20 weeks. So let's talk about hemophilia. So they're hemophilic mice. We did this in collaboration with Dr. David Scott, who's at Uniformed Services University, an expert not only in hemophilia but also in immune tolerance for hemophilia. And what we do here is we give, for the first 5 doses, They get the nanoparticle treatment plus their factor VIII. And then they get factor VIII on this regular schedule that he has for the animals. So in the black line is we just give an empty nanoparticle, and you can see these are antibody levels in micrograms per mil. So these are really high antibody levels, 60 micrograms per mil. If you get a flu shot, you get about 1 microgram per mil of antibodies to your flu vaccines. You get these very high levels of antibody. You can't see it that well. IVIG is just what it is, IV immunoglobulin. It's hard to see a little bit here, but this actually separates from the black line in these first 28 days when you're giving it. So it's a global immune suppressant. It suppresses the immune system, so the animal doesn't make antibodies. But as soon as you stop giving it, it just goes right up to the placebo line. So here in the blue is the SVP treatment. So we give it over 28 days. So now if you look out here, this is out around 200 days, so it's 162 or something days after they've seen a particle. It's the last time they saw a particle. It was 162 days ago. They still have immune tolerance. So we believe we're inducing long-term memory, long-term immune tolerance. We've seen that by profiling the Tregs and other immunological markers. So in terms of antigen specificity, he also then uses sheep red blood cells and phage as well as sort of other antigens. And even out here, these are the SVP groups are responding normally. So we haven't globally immune suppressed these animals. We've taught them to be immunotolerant to factor VIII, but otherwise they're immunologically normal. So we can induce antigen-specific Tregs. One of the key things we've demonstrated is antigen-specific and durable immune tolerance, going out a long time in a mouse, at least at this point. We've stopped injection site reactions, anaphylactic reactions. We've done this with multiple. In fact, I only showed you here. We've done it with over 8 antigens now that we've worked on, both model and actual drugs. We've confirmed the efficacy with David in his lab. He did all that work for us. And we have rapid and robust manufacturing that we've already scaled and gone through and made GMP supplies and completed a phase 1 trial. So that's all I had to say, I think. Yeah.
Thanks for a very interesting talk. It's open for questions now. I'll take the privilege of the first question. Is your concept in the clinic to treat patients after they've developed the antibodies, or to treat them before? I guess that's completely different. So preventive versus you're actually treating the cause. Can you talk about that?
That's a great question. So with the work I've shown here today, it actually only works, and we think it's a good thing, it works prophylactically or co-administered. So one of the reasons we're looking at anti-drug antibodies is I know we're going to get your drug, and I know what the antigen is. And so what the commercial concept would be that if you're going to go on the drug, then for like the first 3 or 4 doses of the drug, you'll get it with the particle. And then you'll just get the drug because you'll be kind of like a vaccine. We don't actually unwind well-established immune responses, which is probably a good thing from a safety perspective. We're not trying to wipe out anything that's up there. We have some other programs that we're looking at to try and do more therapeutic when you have a well-established immune tolerance, but that's less advanced than what I have here.
So my question is very much related, and I think you almost partially answered. The first thing is, how do you induce this tolerance? Because you didn't say. You only said that you have the nanoparticles that they're antigen-specific. Do you put a drug inside, and which one will be the drug?
So as I said at the beginning, one of the keys is we put an immunomodulator inside the particle. So we work with a number of different immunomodulators, and we have a success with a number of them. It's an immunosuppressive drug that's on the market currently. This data I showed you today, and but it has different characteristics when put inside a nanoparticle and given on a weekly basis instead of a daily basis, because the immunosuppressive drug is a global immunosuppressant, and when you go off it, you don't have any tolerance to anything.
But in this case, the antigen-presenting cell will be loaded with the antigen that you provide in the nanoparticle plus whatever is there. So the tolerance cannot be immune antigen-specific, will be more global. So it will be whatever is in the APC, right? Because you are providing, you said, an immunosuppressant to the cell.
So what we're providing to the antigen-presenting cell is an antigen and a signal and a tolerogenic signal. And then that, in turn, induces tolerance long-term. So as I showed you with the factor VIII data, we gave, up days, weekly up through day 28, we gave them the particle and factor VIII. At day 200, they're still getting just factor VIII, and they still have tolerance. So we've educated the body, and it's not a suppressant, it's actually tolerogenic.
And the last thing, and if you add a soluble antigen at the same time,
Sorry?
if you add another unrelated soluble antigen at the same time that you give your nanoparticles, do you have tolerance to see? Because you make it sequential. But if you give your test control at the same time as your nanoparticle, do you induce also tolerance to this unrelated antigen?
Sort of thing. So we do it antigens, like I showed you the KLH and the OVA data, so we do it
together at the beginning. What will happen?
I'm not because the dendritic cells are going to see the immunomodulator in context of that antigen. It'd be more biased towards the antigen of your choice. Yes.
One more question.
Hi. What's your thinking about the autoimmune diseases, where by the time that you see a patient, there may be a large number of autoantibodies? Can you assert tolerance to all of those? How are you approaching that?
Yeah, so we've made some reasonable progress in our JDRF collaboration on type 1 diabetes. The main reason we're focused on anti-drug antibodies is in the autoimmune disease, it's not clear what the antigen is. So in type 1 diabetes, it could be insulin, it could be chromogen, it could be GAD. So at least from our first foray into this, we see the anti-drug antibodies, I know we're going to get the drug, and I know what the antigen is. So that's what we see. But we continue our work with JDRF and in other autoimmune diseases. And we have some other work that is, we didn't talk about today, more focused on therapeutic tolerance.
You mentioned about 30% of patients may be developing anti-drug antibodies. Do you have a way to find these patients or select these patients ahead of time so you don't expose 70% of the patients to something that's not needed?
So it depends on the drug. So in hemophilia, about 30% of the patients make inhibitors. And Humira turns out to be 28%. With the uricase drug, it's 92% of the patients. So it depends really how foreign the drug is to that person. There is some predictive markers, but not well established. There are cases in the hemophilia where they start seeing antibodies and they go down. So it's not like you start seeing them; they always go up. But yeah, that's something we're certainly looking at. If we had a better predictor, it would be good.
So we keep talking about nanotechnology, and we're talking about putting drugs in, and usually the drugs are a aggregate of a drug. It's not an individual molecule. Have you been able to pull apart the individual molecule versus the aggregate of the drug and the difference of the immunomodulatory effect of those two things? This is not a simple question.
So the immunomodulator is a small molecule, so I don't know that it's an aggregate.
No, everything from everything I've seen, and Obed, please correct me if I'm wrong, you usually have an aggregate of things, not one molecule, but say 1,000, 10,000, 100,000 molecules. It's not going to be one molecule. It may see one molecule on an aggregate, and it may be presented with just that one piece, but it's not going to be one molecule seeing one thing. And has anyone done this? Has anyone looked at the individual molecule, not the aggregate and the surface of the aggregate that's interacting? Do you understand what I mean? It's not a fair question for specifically you.
But
Yeah, I don't.
all right. Thank the speaker once again. So our next